Thank you very much, Matt, for agreeing to give a Build a Cell seminar, and you can get started.
All right, thanks, Kate. Hi, everyone. I'm Matt Good. I'm an associate professor in cell and developmental biology at the University of Pennsylvania. I'm always excited to give a talk for this community, which is really, dear to my heart. And one of my students will be at the Build a Cell workshop next Saturday. And I should say, I'm going to talk about a couple stories from the lab that we have under revision. And so there'll be a couple breaks throughout. So please feel free if you have questions to put them in the chat, and I'll look at them as we go along, or we can take a break during. Okay, so my lab's interested in the assembly of synthetic cells and synthetic organelles. On the left, this includes bottom-up assembly of synthetic cellate compartments that contain, for example, cell-free cytoplasm that can carry out aspects of the cell cycle, such as spindle assembly, microtubule patterning. And then we've also been interested on the right in kind of top-down approaches to augment the function of living cells by embedding within them synthetic organelles. And this is really the focus of my talk today. Okay. So, particularly for this community, I don't have to explain, there's a number of hurdles towards reconstituting cell-like systems, and my lab's work to address a number of these challenges. Many of us work with vesicle-based systems. Rigid membranes really work well for drug delivery, but the synthetic cell needs durable and dynamic membranes, for example, capable of embedding proteins, interfacing with other protocells or living cells, for example, to engulf them. I like to show this movie from the Emmenegger lab of vesicle engulfing. This is a dendromersome engulfing a bacteria. And then these need to also be responsive to external cues. For example, we've developed light responsive lipids so that we can get this partial disassembly or fission of vesicles in response to light. And then to be able to make functional minimal cells, in our view, it's really necessary to reliably load components without dilution, particularly complex mixtures such as the cytosol. This is really kind of something that I started in my postdoc work with Rebecca Heald and Dan Fletcher. It's a really challenging problem. We've previously tried to approach this by using microfluidics to encapsulate oocyte cytoplasm without dilution, for example, in water and oil emulsions. And then we've been working with Dae-Yoon Lee's lab at Penn to use double emulsions to create these compartments as well. And then, of course, to carry out reactions selectively, to really build cell functionality, we need to be able to subcompartmentalize encapsulated components. And this is really what I'm going to focus today's talk on. And so we've largely investigated protein-based compartments or condensates as a strategy for compartmentalization. So as many of you know, coacervates and condensates are broadly useful to the synthetic cell community as materials to promote reactive separation. These compartments are often formed, for example, on the left, building these compartments in vitro from polyelectrolyte polymers. But for today, and what my lab focuses on is using peptides or proteins, which offer this additional benefit that they're a polymer. You can make coacervates, but they can also be genetically encoded so that they can be expressed in living cells so that you can self-assemble these materials or these compartments. And the same is true in protocells, for example, with transcription translation extracts. So, we've of course taken inspiration on how to build compartments from natural membraneless organelles, such as the nucleolus and stress granules. The cell contains dozens of these compartments that self-assemble, for example, from proteins and RNAs into these micron scale assemblies, and they do so in the absence of a lipid bilayer membrane. And commonly the proteins shown on the left here contain large disordered regions. Commonly, proteins that facilitate these assemblies contain large disordered regions to create multivalent interactions and really drive this condensation. So the key here is just multivalency. I'm not going to spend much time on this. You can achieve multivalency a number of different ways. So many signaling proteins contain multiple folded domains and will bind to short peptides or linear interaction motifs on other sequences, like in the case of NCK and NROS. You can also engineer your own multivalency, for example, by using multiple SUMO domains, which bind to multiple SIMs. These generate really nice liquid condensates. But what we've really focused on and what's more common in biology is the assembly of low complexity sequences, essentially unfolded polypeptides that have these disordered stretches. And the multivalency comes from interactions often considered weak along this disordered chain. So just for context, unlike how we think about assembling rigid protein compartments, if you think about nanocages or viral capsids, we understand the assembly of membraneless organelles is really driven by equilibrium phase separation. So disordered polypeptides went above their critical concentration. If you're looking at a test tube, you'll get this self-assembly into condensates that visually look like a turbid solution at the macro scale, and then these liquid-like droplets at the microscale. And just like traditional organelles, I think many of you already know this stuff, but these compartments have specific biochemistries and also general modes of action. And so you can read many different reviews on this. Instead of highlighting like the 10 ways that protein condensates control functionality in the cell, I just want to highlight two very simple modes of action. So on the left, these compartments can function as insulators by sequestering components. So if you think about reactants together carrying out a reaction, the rate of this reaction can be strongly inhibited by sequestering the components away from one another, effectively lowering their effective concentration. This is, for example, how stress granules work to separate elongation initiation factors and RNAs that are no longer translating. So this is one style, right? Many condensates will functionally segregate or separate components. And then the other mode is that these compartments often are enhancing the local concentration of components such that they accelerate reactions, and we often describe these as bioreactors. All right, so over the last, I'd say 7 years, there's really been an explosion in interest in engineering condensates to control and augment cell functions. This is also true in the protocell community. And so, for example, I've highlighted here examples of controlling metabolic flux, kinase transcription factor, signaling, connecting components that would normally phosphorylate one another. And then this kind of insulator idea on the lower right, which is something that I'll highlight a little bit today. And then, to get you more excited, along with these studies have come lots of new IP companies interested in modulating both the properties of condensates and their functions in cells for a variety of biomedical applications, in addition to just more standard synthetic cell applications. Okay, so as a preface, I kind of want to explain what I think are really essential questions and pitch a potential approach. So in thinking about a protocell or synthetic cell, what are really the features that would be useful? And I think a key point is restricted permeability, this idea of compartmentalized components such that you don't have free diffusion among these discrete subcompartments. Importantly, we would like to have some type of spatial and temporal regulation of this assembly or the separation, segregation of components. And then I'll make the pitch that I think it's really important that we want to be able to coordinate these modules. So integrating them together to create kind of daisy chains of chemical functionality. And this is often quite challenging to do completely from the bottom up. And so our approach has basically been to try to individually characterize a synthetic organelle by proof of concept testing it in extant cells, in living cells, and testing whether we can augment their function with the idea that this could then be ported back into a more simplified cytoplasm in a protocell. Okay. So one other piece of background about my lab and thinking about how do we approach this engineering challenge. So one side of the lab has really been focused on biophysics, materials, properties of these condensates. So for example, what are the sequence rules of these disordered chains that govern their self-assembly? Can we tune CSAT, material properties, et cetera? The second point, I'll tell you a story that came out last year that I've been trying to understand how to create distinct compartments that don't co-partition with one another, so how to achieve selective condensation. And then trying to engineer the interfaces. So I said there's no membrane around these compartments, but there are surfactant proteins that exist in nature or that you can build that will coat these interfaces and allow for control of size topology of condensates. All right, so that's one side. On the other side, we've really been focused on kind of more traditional synthetic biology and how to integrate these compartments, these condensates, as protein switches, as a way to control cell functionality. I'll go into this in a little bit more detail, but essentially, we can genetically deliver a protein of interest, in this case, like a disordered protein scaffold, CRISPR tag clients, and then use this to selectively compartmentalize them. And on the lower right, kind of the last new study from the lab that's a preprint is how to do this non-genetically. So can we actually build these compartments first and then deliver them to cells? All right. So instead of going over a lot of the individual studies, I think what I want to summarize is that we've built a toolkit for programmable assembly of condensates and as well as targeting and release from them. And I can walk through this. Let's see. Can you see my cursor? Great. All right. So starting from individual polypeptides or proteins, we've tried to understand sequence rules that govern this self-assembly, how to tune saturation, concentration, et cetera. Once you have the compartment, to achieve selective targeting, we've really focused on using nanobodies or originally small coiled coils that have nanomolar affinity for one another, and then use these to enrich or sequester targets within these preformed compartments. And then because we want to use these as protein switches, we'd like to have dynamical control both of assembly as well as release. And so we've developed a number of strategies to then liberate these components or release them from the compartments through optical thermoenzymatic means. And this toolkit was built over a series of papers, which I've listed here, and others have also developed similar toolkits, but really what this is meant to demonstrate is feasibility. So we really have tools now that we can build synthetic organelles and target components on demand. Okay, so I want to go through one proof of concept, a study from 2021, where we sought to encode synthetic organelles and use them to target any enzyme or pathway of interest. And the approach, which I mentioned in the last slide, but I'll really want to codify here, we have a scaffold, which is something that can self-assemble to make a compartment. And a compartment, I mean a mesoscale condensate. So a partition space inside the cell or the protocell that, we're typically in the 1 to 2 micron diameter range. So the size of a traditional organelle. So you can deliver this gene by a number of different strategies, viral delivery to encode it in the genome. And then we'll target individual clients inside the cell. So we just CRISPR tag their C-terminus using these short coil coils, the SynZips from the Keating lab, or some additional coil coils that Roman Girola's group has developed and characterized. And so doing so by expressing the scaffold in the cell, and then by having this tag on the native client, you can actually get these to dimerize, and this will lead to selective partitioning of your target of interest into the compartment. And so the purpose of this study was really a proof of concept. Can we get this inducible recruitment, sequestration? We try to build an insulator, which is what I'll tell you about, and then dynamically control the release. So really to use these as a dynamic switch. So for this study, our goal was essentially to take a simplified model cell system, in this case budding yeast, and to optimize scaffold and coil to achieve selective partitioning or sequestration of the organelle. So I'm showing two columns here. One, so in this case, we have scaffolds that have different length of the IDRs. In this case, this is 3 repeats of 1 IDP. And in green, you can see the compartments form. And depending on the type of coil that you use, we're going to have multiple compartments per cell, or 1 large compartment per cell. So scaffolds in green, and then our targets in red. So we can get very strong partitioning of the target to this compartment, this condensed phase, by using a coil. And I'm highlighting the numbers up here. The quantitation is shown throughout the slide, but basically by lowering CSAT enough and relative to the level of expression, and by using these high affinity coils, we can get 95% of the scaffold is partitioned in the condensed phase, meaning only 5% of it is sitting around in the cytosol. And then we can get over 90% of a target sequestered. And this is really important for the idea of an insulator, because we want to create a functional knockdown. So we're pulling proteins away from their normal cytosolic position or their nuclear enrichment, and sequestering them into these condensates. Okay, so we targeted 5 different enzymes that we thought would be essential for cell processes like proliferation, regulation of the actin cytoskeleton, et cetera. I want to just highlight 2 here for control of the cell cycle control system. So essentially 2 enzymes, a GEF, CDC24, it's a GEF for CDC42, and then CDC5, which is a poloid kinase. So these control or essential for entry and exit from the cell cycle in this model system. And so I'm showing data here for targeting CDC24. Normally, there's about 40 to 50% of cells that are arrested or unbudded. If you just express the scaffold, nothing happens. If you just tag the client, nothing happens. But if you put these together, then you can get a very strong increase in the amount of arrested cells. If you follow them over time, we can see that we can completely shut down proliferation. So as a proof of concept, this was working. If we take key enzymes and we target them to the synthetic organelle, then we can actually shut down those pathways. So it does function as an insulator. But of course, our goal was to go beyond just constitutive insulation. So we really wanted to have reversible or dynamic control over the pathways. I'll show you 2 strategies that once we sequester the component, how to actually release these on demand and restore functionality. So we developed 2 strategies. The first I'll show you here is an optogenetic one. We used a photocleavable protein on the scaffold between the IDR and then this sequence that this coil that's recruiting the target. And so then by pulsing the system with light, you can get release from the condensate or the compartment on the order of, shown here is 5 minutes. So this can be relatively fast and dynamic. And then to show kind of phenotypically what's happening, the time scale is much longer. We're looking at cells arresting or re-entering the cell cycle. So at time 0, we have about half the cells arrested. And we're going to add an inducer to express this disordered scaffold protein that will form the compartment. And so this will then sequester the native CDC24, and it's going to shut down cell growth. And so that's what's happening over time in this gray curve. And if you let it keep going, you reach over 90% of the cells are arrested. But if we wait 4 hours and we're starting to get a lot of arrest, if we pulse the cells with light, then we can reverse this process. And again, it's because we're liberating this essential enzyme that we've sequestered. So this works. This is essentially an irreversible process, but as the protein gets re-expressed with the tag, in theory, you would need to pulse the system again. And then we also tested a reversible strategy, which was to use, again, coils, but ones whose interaction is dependent on temperature, so that they can be dimerized, for example, at 25 degrees at room temperature. But if we raise it to 40 or 42 degrees, then we can dissociate these interactions. And I flipped the axes, but essentially, again, we're starting from about 50%. This is like 1 over arrest. If we express the scaffold, the compartment forms and we'll start sequestering CDC24 in arrest. If we change the temperature, we can liberate CDC24 again. And then if we cool the system, we can start arresting again. So as a proof of concept, it's absolutely feasible to generate synthetic organelles in cells that function as insulators, and you can use them to target and sequester enzymes in native signaling pathways. So I'll pause just in a second. I just want to give one last slide here on the section that I showed you, use of this in a simplified model like yeast. Many other labs have done this, similar things, for example, in E. coli. Our goal in part was to try to do this for mammalian cells as well. And so I'll just show you that we've developed a lot of the same tools here so that you can get inducible expression of the scaffold to form these compartments. By virally delivering them, you can generate stable lines that will express these. You can have them be cytosolic or nuclear. This includes primary cells. We've been focused on trying to do this in immune cells. And then Astute and Wental has the photocleavage system working in the human cells as well. So at least for the kind of roadmap for today's talk, I think the important thing is that we figured out a way to take our knowledge of the biophysics and materials in vitro and show that we can put these proteins, make synthetic condensates in exon cells, and then use them to control cell decision making. And so in theory, you could do the same thing in a protocell. All right, so I'm going to pause for a minute and just look at some of the questions in the chat. Okay, so one of the first questions in the chat is about, in terms of forming compartments, do you consider use of other polymers to make artificial compartments as well? Of course. And I apologize if I didn't make this more clear at the beginning. In fact, most labs who are building coacerates are using polyelectrolyte polymers. You can use basically any 2 components that have opposite charge. So you can take like supercharged GFP as well as another multivalent polymer, or just many of the polyvinyls have traditionally been used. I want to highlight the reason that we've really been focused on peptides and proteins is that we can go beyond this simple characterization, outside of a cell and actually get these components to be made inside of a cell. And at least for the synthetic cell, the protocell community, our idea is that it's the same thing if you wanted to build a compartment in a transcription translation cell-free system, right? So if you had a vesicle, TXTL, you could include a plasmid for this protein, and it would self-assemble to make a compartment. So that's really been our focus, the reason we focused on peptides and proteins. Okay, next question relates to reversible or released from the compartments. And the question is whether there's specific control over the size of the assemblies, how big the organelles are. So it's a good question. I made one note that by changing homotypic strength among the coils, this is actually what we're using to target these native components. But if you increase their propensity to interact with themselves, we can kind of shift from having many smaller foci to one larger foci, one focus in the cell, one compartment. What I'm going to tell you about in the next part is actually another strategy to regulate size through using surfactant proteins. So maybe I can come back to that. All right, and then another question about reversible is how fast can it be? Right, so it depends on the type of tool you're using. We've tried enzymatic cleavage with proteases such as TEV. This is a common way to build protein-based switches in synthetic biology, is that you use a protease to cleave off a recruitment tag, a localization tag, et cetera. And the kinetics of that depend on the concentration of your enzyme. So in vitro, we can get release kinetics with enzymes like proteases on the order of tens of minutes. In the cell, it's a bit slower. In terms of the thermal reversibility, this is very fast. And so because these compartments are not actually gels, they're liquid-like, then if you do photo recovery, if you look at diffusion of the scaffold itself, the turnover rates can be on the order of seconds. So in the scaffold I showed you, this triple LAF1 RGG, the kinetics we measured with FRAP in vitro, whereas the half-life, the half-time of recovery was on the order of 5 seconds. And it was even slightly faster in the yeast, like 3 to 4 seconds. So there's a very dynamic turnover of the scaffold. So what this means then is whatever you do to, for example, cleave off a recruitment tag can actually lead to very fast dynamics for either recruitment or release. So for thermal release in vitro, again, this can be on the order of seconds. What we showed in the yeast system was phenotypic kinetics, which for the cell cycle were on the order of hours, but we also looked at actin patches and cables and their polymerization. And so I would say, in short, it comes down to, are you looking phenotypically, or if you just want to have release of the component from the compartment? And if it's the latter, then it's absolutely on the order of tens of seconds to minutes. Okay, I think that's it for the chat right now. So, right, just to reorient, in this first part, I told you about proof of concept. We can use these synthetic organelles to control cell decision-making. In the next part, I'd like to explain a couple stories that you can read about from our lab on how to build discrete compartments, spatially discrete compartments, multi-phase assemblies, and then the idea of trying to do this non-genetically as well. Okay, so for the second part, I'm going to highlight work, a study that came out last year from Rachel Wells, who was a student in the lab. And her goal is really to identify different types of peptides that are capable of forming discrete condensed phases. So the way we started this, Rachel wanted to do kind of a preliminary screen, and she and some previous members of the lab, by basically doing a co-expression screen. So the idea is to take different disordered proteins, fuse them to GFP or M scarlet, and then co-express them in a really simplified eukaryotic cytosol, that of budding yeast. We could have done this in protocells. We could have done this in human cells. We chose to kind of pick yeast because we used mostly human proteins. And the goal was really to break what would be the natural interaction networks to really focus in on specific features that, or things specific to the IDRs in terms of their ability to assemble, either co-assemble or form discrete compartments. So here's an example of some of these proteins. We picked ones that were known to be able to form condensates when overexpressed in cells, and that had, in most cases, had a fair amount of biophysical characterization. And so what we saw was mostly the bottom panel here. You take two full-length IDPs and co-express them with these fluorescent proteins, and they'll tend to co-assemble. There'll be almost perfect overlap among them. But in 3 cases, we found pairs of peptides that formed these discrete red and green compartments when co-expressed at the same time in the cell. And one thing I want to highlight is that you may think that these would, if you know aqueous two-phase systems, that you might think, oh, this is all PEG dextran. We're going to find very different chemistries in these polymers. But actually, most of these proteins have large disordered stretches that contain the normal complement of amino acids. So we didn't find one that was all aliphatics and another that was really charged, and these did not co-assemble. In fact, the ones that we found, I think by primary sequence or by sequence composition, you wouldn't necessarily pick out as being unlikely to form discrete condensed phases. So ultimately, in the paper, we decided to focus on one pair that uses the low complexity domain from FUS and the RGG domain from LAF1. We had some additional work from a collaborator, Jitain Mittal's group, simulations that suggested that these were miscible. And then what did the actual data look like here in yeast? So we're expressing, if we take this top panel, LAF1 as a tandem, it makes these green condensates. The low complexity domain of FUS LC is what I'll call it, has a very high CSAT. So it doesn't, with a single domain fused to a fluorophore, it doesn't form compartments. So it's really the client here. And what we could just measure was how much is that client partitioning to our LAF1 condensates. And the answer is almost none. The enrichment index is right around 1. So it was not partitioning, but this didn't prove that it was able to form separate compartments. So we multimerized it using 3 of these low complexity domains. And then in magenta, you can see it's able to form compartments. And if we look at the merge, these magenta compartments are different than the green compartments. So this is a pair that doesn't want to co-assemble with one another in this context in the cytosol. But to make sure this was really something that we could understand, Rachel purified the components, Rachel along with another postdoc in the lab, and was able to show very selective partitioning of, in this case, we're forming LAF1 condensates and looking at LAF1 partitioning versus FUS partitioning to these preformed compartments. And so this is important because it says you don't need cytosol, you don't need RNA. It's really something hard-coded into these polypeptide sequences. And this gave us some confidence that we might be able to dig deeper into what's controlling the selectivity. So this is kind of a summary slide. And what I want to say is that you can purify these proteins and make them form beautiful compartments in vitro. They will never co-assemble with one another. You can also express them in other contexts, like in human cells, they'll form discrete compartments. And then what's important is we tried many, many different types of mutations, and we were able to find ones that actually would cause blending. So we could make mutations to the sequence of FUS, and now in vitro get full overlap of this, so full co-assembly with LAF1, and the same thing in yeast, in human cells, et cetera. And so these results seem portable, but really the conclusion from this is that there's sets of IDPs or IDRs that have the ability to form discrete condensed phases, and then this is dependent on features of their sequence that we could tune. And actually, one of the takeaways from the story, it was particularly beguiling for a number of years, is that it's really a balance of homotypic and heterotypic interactions that controls this selectivity. All right, so I'll try to explain this. Homotypic is one polymer, like in green, interacting with itself. And heterotypic would be green interacting with magenta. And so what was really surprising was that we tried many dozens of mutations where we added residues to FUS that we thought would push its heterotypic interactions, cause it to interact more with LAF1. And in essentially 99% of the cases, it did not. And the reason that we were able to deduce was because it was actually strengthening its interactions for itself. And so as a follow-up to this story, we've been working on the idea that if we actually weaken homotypic interactions, we can take 2 polypeptides that normally aren't co-assembling and then get them to assemble. So there's not something radically different about the chemistry of their sequences, but it's this strength of this homotypic interaction that is what is preventing the two from co-assembling with one another. And this is what another Rachel has been working on as a follow-up. Okay. So second kind of summary of this roadmap, we've identified 3 sets of IDPs that form spatially discrete compartments. Other labs have done this with factors that form like mediator complex that form nuclear foci. And there's also a nice screen from Pilong Lee's lab that is further getting this. This is a really fundamental question. How do IDPs partition into the correct subcompartments? And for us, for the synthetic cell community, we think it's really important that we can take pairs of IDPs that will self-assemble to make these compartments and that they won't necessarily mix with one another. Okay, I'm going to answer some other questions from the chat before moving on. So one question is, can FUS carry payloads? And I think this, you're asking, can the FUS compartments carry payloads? And so the answer is absolutely yes. And so you can do this a couple different ways. You can use coils like the ones that we've used to then target another enzyme with that IDP to partition to the condensed phase. Other groups have also used the actual sequence of FUS itself and just fused that, the low complexity sequence, to an enzyme. So you can get this partitioning, this multimerization and partitioning using the IDR. We don't think that that's quite as effective, which is why we usually dimerize the target and the scaffold, and that gives really good partitioning. So yes, you can, and I want to say, you know, we want these to be orthogonal, but the reality is they're also partitioning other factors. But the level of partitioning tends to be quite low. And then we want to drive much higher selective, stronger partitioning by using a nanobody, coiled coils, etc. All right. Another question from Adam Arkin. Rules guiding, yeah. So what are the rules guiding payload size, hydrophobicity, or can anything basically be a payload? Yeah, so we haven't, so one, we've spent a lot of time thinking about this, working on this. We've seen some size dependence. So if you have, you're dimerizing a coiled coil to the scaffold to get partitioning, the size of that target, the longer it gets, particularly if it's a folded protein. If you just think about having an IDR and a folded protein fused together, the relative amount composition of each of those will control CSAT. So as you recruit larger and larger amounts of bigger proteins, then you can actually start to raise CSAT and get smaller, and that will affect foci formation. The way we deal with this is to control the stoichiometry. So we try to have the scaffold be 10 to 20x higher than the target. And then the partitioning of the target will only lead to like a small, very small shift in CSAT. In terms of the composition, whether it can be, you know, electrophilic, whether it's hydrophobic, et cetera, we haven't seen any clear rules for that. But again, we're just dimerizing a client to a scaffold, and that represents, you know, 5% of the stoichiometry of the overall scaffold. So in that case, it's relatively insensitive. Other groups have explored this more extensively, and certainly there are additional partitioning that occurs based on the actual chemistry of the scaffold. See if there's anything else. All right, so the next question is actually leaning forward to multi-phase assemblies, which I'll get to in a second. And, oh, sorry, one last point, Adam, or can we combine these peptide-based coacervates with membranes somehow to have IDPs make their own membrane, and can they do things like keep ion gradients? Lots of really good questions. Okay, so yes, you can absolutely get ion partitioning, and you can maintain pH gradients. So in the condensed phase, actually the scaffold, the polymer here, its concentration starts to be high enough that it can compete with the surrounding solvent for proton exchange. And so a number of groups have papers on bioRxiv showing, or published work showing, that you can create pH gradients, for example, in the nucleolus, or even just from an individual IDP, making a compartment, you can hold 1 unit pH gradient. So that's a 10-fold difference in the proton concentration. In terms of using actual membranes around these, there's a bit less on lipid bilayer-based membranes, but people have worked on a variety of types of amphiphiles. So there's just a paper last month in Nature Chemistry of trying to use polyvinyls as ways to coat the surface of these compartments, for example, to stabilize them or prevent fusion. I think this is a really exciting area. I will note in general, those are not going to be necessarily portable into the cell. And so what I'm going to talk about in the next section is how to create skins or surfactants that will partition to the surface that can also be genetically encoded. Okay, so hopefully I got to all the questions. So let's move on. All right, so we've been really interested in how to control these interfaces and assemble multi-phase condensates, and using a lot of the same tools that I told you about. So importantly, we've been focusing on the idea of designing surface active agents from polypeptides. Surfactant proteins lower surface tension and can prevent fusion. And so you can actually use them to control the size or topology of client recruitment to these compartments. So what a previous postdoc did, he made these RGG-based condensates, which are shown in green. And then he built tri-block or quad-block surfactant like molecules, where we have a disordered domain and then additional folded domains, including a fluorophore. And we want to control the balance of partitioning, right? The more disordered sequence, the more it wants to go into the condensed phase. The more of these folded domains, the more they want to stay in the continuous phase or with the solvent outside of the condensed phase. And so by testing a couple dozen different domain strategy or structures, organization, Ben initially found a number of candidates that would strongly partition to the interface. That's shown here in red. And then by controlling the stoichiometry, by increasing the amount of surfactant, that you can start with droplets that are quite large and actually dial down the sizes. And so this is a really important feature of using surfactants. The other way we wanted to use this was to control the topology of recruitment. So Mo Yang, a student that I've co-mentored, who, along with the collaborator Dan Hammer, was interested in taking a very similar type of strategy, but simply adding a coiled coil. And so in this case, the surfactant is green. We have droplets shown in bright field. The surfactant enriches at the interface. And then because he's using these coiled coils, he can then get the cargo to be targeted to the surface rather than the interior. And so we can now enrich the target of interest inside the condensed phase or on the surface. And we think this is useful for spatially organizing biochemical reactions. So really, the story I want to highlight from Wu Yang, that's on bioRxiv right now, is his interest in using these surfactants to actually wet the interface of two condensates that would normally co-assemble with one another. And so these are our familiar friends here. This is from that the paper last year, the Nature Chemistry paper, a pair of these IDPs that self-assemble to form discrete condensed phases that's shown in this image. So they can actually wet to one another, and they will come apart, but they will never co-assemble. But he tested a small library of tri-block surfactants where you have now two different arms. One is the disordered sequence of one of the factors and the other disordered sequence of the other IDP. And then again, a small library of different folded domains. And by doing so, he was able to find one of these tri-blocks that was very effective at wetting the interfaces so that you get this co-assembly. So now the internal phase here is FUS, and the exterior of this is LAF1-RGG. And again, this will never occur without these surfactants. And then additionally, because the surfactant itself contains these disordered stretches, then it has a saturation concentration. And so by changing the relative stoichiometries of these components, he could create these triphase assemblies where we have, again, FUS-LC in blue, RGG in green, and then in magenta, this surfactant. And so we're really excited about this just as a tool. These scaffolds are not targeting anything right now, but if we include coils on them, then we can, in theory, partition selectively into these compartments. In this case, 3 different compartments, 3 different targets of interest. And again, I'll highlight this work is on bioRxiv. Now, I want to be clear about one thing. This is not just something you can get from any surfactant. So if you just try traditional small molecule surfactants, you don't get this effect. You really need to have these much longer polypeptides. There's some works from other groups that suggest that you might not actually need polypeptides. You can use other polymers to jam these interfaces as a way to promote or to get wetting and promote assembly. We haven't gotten that to work yet. And then I also want to highlight that in some cases, you don't actually need surfactants to get interesting structures. So one way we trigger the assembly, the self-assembly here, is that we include a solubilization domain on one of the individual polymers. In this case, it's on FUS. And so it's fully soluble until we add a protease to remove that tag and liberate the assembly. And if we do this while we're also including the other IDP, and we do this quickly, we can actually get these multi-phase assemblies where we have droplets of FUS inside RGG, or even these kind of chains of droplets together that create this like caterpillar-like shape. And so the surfactants are really what we want to move forward with, but of course, many of these interesting structures can also be created by kinetic trapping. Okay, so for this section, why is this work significant? In the previous section, I showed you we can build discrete compartments from different IDPs, right? We can make like red and green compartments in the cell or in vitro. And then in this part, I've shown you that we can use or build surfactant proteins to create core shell topologies or these multi-phase, biphase, or triphase assemblies. And we really think this is useful to construct minimal condensates that can be used to spatially partition biochemical reactants. Okay, so maybe I'll just check if there's any other questions. No, okay. All right, so for the last part, I want to talk about, I'll fully pivot. I've told you everything that's great about polypeptides is that we can deliver them to cells, right? We can use a virus, get the gene into the cell to encode this peptide, and then the peptide will self-assemble to make the compartment. And what one of my students, Bobby, was interested in, and he'll be at the Build a Cell workshop on Saturday. So definitely go talk to him if you're going to be there as well. Was can we actually build these compartments outside the cell and then deliver them to the cell without having to use genes? So we were inspired by work from Ali Mizraz's group, who identified essentially short pentamer peptide sequences such that you can make like a 26 amino acid peptide that will form a coacervate. It'll self-assemble to make a particle. And these have the unique property of being able to cross the plasma membrane. And so this is an image from Bobby himself of taking some of these HB-PEP coacervates, form those in vitro, and then add them to cell media. And this is U2S cell that contains perhaps 100 of these compartments that were made without having to encode the gene. And this is the nucleus in the middle, and this cell will continue to divide. Again, this is not genetically encoded, so they won't propagate with the cell division, but we can load up a cell with these pre-made compartments. And then Bobby, just as a highlight, Bobby was also interested in moving away from having to CRISPR tag targets, so he wanted to try a strategy where we would include nanobodies in these compartments, and then this would really create a reaction hub for interacting with native signaling components, but again, pre-assembled in the lab and then delivered into the cell media. So this is pretty straightforward. by increasing the concentration of the peptide and using a pH shift, you can promote assembly of these particles, these coacervates. These are essentially gel-like condensates. And their number and size depends on concentration, of course. We were interested in making ones that were 2 to 5 microns in diameter, have a relatively large hub inside the cell. Oops. If you photobleach them, fluorescent version of the peptide, you can see that there's essentially no recovery over 5 minutes. They really are gels. This is something we expected. And so how do you get stuff inside, right? Ollie's lab had really nice work showing that you can pre-assemble these with RNA or protein. And the way this works is you just include it in the solution prior to the pH shift, and then it'll actually get trapped inside these gel-like coacervates. And so that's on the top row here. So you can get non-selective, nice loading inside of them. And what Bobby wanted to do was increase the selectivity or the overall level of partitioning. So he included some of the peptide sequence onto his target in this case, which is GFP, and was able to show he could increase the loading, right? So this is just co-assembly of a target of interest with the compartment. Of course, this is not what our main focus is. We really want to create hubs to interact with other factors. So then Bobby co-assembled these with a nanobody. In this case, it's VHH4, which is a very high affinity for GFP, which is super useful, right? Many of us GFP tag our targets. So in the left column in red is the nanobody or just M-cherry alone. So the top row is just our control. And then VHH4 with the HP PEP attached. So here's the particles, and then this is target recruitment in green. And so what you can see is we get no recruitment to the control, and then nice recruitment to the surface of these HP PEP particles using VHH4 that's quantified on the right. So this works in vitro. He wanted to test it in cells. So the same thing on the top row, we have particles delivered, but without the nanobody, just M-cherry in them. And if you can see, we actually have exclusion in most cases from the particles loaded in the cytoplasm of the cell. And that's shown here. It's the partition coefficient on average is about 0.7, 0.8. So we're getting mostly exclusion. And then when you include the nanobody, then you can get nice, strong enrichment, which is quantified here. So it looks like the system can work. What we're really trying to optimize now is the efficiency of release. So these actually get taken up by macropinocytosis, which means they end up in an endosomal compartment. And we use TADHA2 peptides or chloroquine to release them to basically deacidify endosomes or to create holes in the endosome so that they can lose the membrane around them. And then they're this free-floating hub. And so this is a dated figure now, but we can get up to about 25%. Still not perfect, right? We're not at 100%, but of particles will be released from the endosome and recruit the target of interest. Okay, let's see. What's the pH stability of these? Do they have biochemical advantage over other things? Yeah, so the pH dependency, in this case, the original HP PEP that the Misra's lab characterized, we store the peptide at pH 4.5, and then we shift to pH 7.5, and then they gel. However, they can be reversed. So if you shift the pH back down quite low, then you can start to get disassembly. And this is may have been intentional or not, but this is actually somewhat useful if you want to have client release from an endosome, right? If the endosome is acidifying, then you might start to get some disassembly of these peptides or these peptide coacervates. In our case, we actually don't want that, and I'm not showing it to you here, but we measured stability over time, and we show that if the particles were 2 microns or larger, that we could get basically no degradation of these compartments over the course of 5 days. And so what we'd love is to be able to preload these compartments with enzymes of interest or with nanobodies, and then actually have them stay. But if you'd like to learn more about the pH-dependent release, or you, in the case of this other peptide, you can get glutathione-based reduction, increasing the CSAT, which leads to partial disassembly, then I'd point you to papers from Ali Mizras's group. There was also a question about general is to have distinct types of memberless compartments in synthetic cells, some sequestering different payloads. Yeah, so absolutely. Maybe I'll just go back to this general slide here. We can absolutely create the distinct compartments. And then earlier in the talk, I told you how we can target a factor of interest to that condensed phase. And so we used coiled coils, one on the scaffold, one on the target, and you get very good partition specificity. In the latter part of the talk, in the last few slides, I showed you how we can use nanobodies to do that. So it really comes down to what you want to target. But let's say you wanted to have a red and a green distinct compartment inside a protocell. You could use these two peptides, FUS-LC and LAF1-RGG. And then you could use coiled coils, which I'm happy to talk with you about offline, but there's a large panel from the Keating lab, as well as another 6 well-characterized from Roman Girola's group. And so these are orthogonal pairs of these coiled coils. And so you put one on your scaffold and the other on the thing you want to target. So for a TXTL, you'd be putting in plasmids for both scaffolds. And then the question is, what would you want to target? And if it's something new you also want to make, then you would have a plasmid for that. Or if you wanted it to be a native E. coli protein to sequester some aspect of transcription translation, then you would need to have that factor targeting either through the coiled coil or an antibody. Okay. Yeah, and the last question was about what determines whether you end up at the surface or go on the inside. In this case, this is how to create an amphiphile. Okay, so there's a lot of theory behind this, and we don't have the ability to predict this for the proteins. We're working with a group here at Penn, Rob Phillips, to try to have better design rules for these peptide-based surfactants. But our strategy is basically to create a small library. So we use the arms make the arms from the individual disordered sequences. And then we use basically GFP, mCherry, BFP, as well as other folded domains like typical that you would use for purification, GST, MBP, SUMO, et cetera. Okay, and so the more you put in the middle block, folded domains that don't necessarily want to partition or enrich in that condensed phase, the more likely they want to stay in the continuous phase. That creates this amphiphile effect where one part of the molecule wants to be in the continuous phase and the other part wants to be in the condensed phase. Now I'm showing you a more complex example here. We're trying to create these multi-phases, but again, I can just go back a few slides. So if we, in this case, there's a GST and an mCherry on the surfactant, and they do not want to partition. When we measure their enrichment index alone, it's around 1. So they're not excluded, but they don't want to necessarily enriched in the RGG condensed phase. And then the other arms on this surfactant are the RGG, this disordered sequence, and that does want to partition. And that's how you create the amphiphile. But the key point is we can't predict this. So we had a number of really beautiful candidates from the library. And when I say a library, I mean we tried like 12 constructs and purified them, Ben purified them. And some of them work really well and some of them don't. So we don't know the rules, but I think if you had your condensate of interest and you wanted to make a surfactant, I think you could probably do it by making 12 constructs, and one of those 12 would work very well. Okay, question about these kinetic trap structures. So the answer here is you don't need the surfactant. So the way this works is that if you get very rapid assembly of FUS, then it will actually it'll be inside the RGG condensed phase such that it cannot kinetically move out of that phase. And so then energetically it's stuck inside the other compartment. In this case, there's not one trapped inside the other. It's that their interfaces, again, FUS in blue, RGG in red, are trapping the proteins in a way. This is partly a unique feature of FUS-LC. So it has this feature that it likes to rapidly age. So over the course of tens of minutes, then these will start to become more viscous. And this contributes to this kinetic trapping. But I want to point out this has been well described for other types of polymers as well, and relates to essentially the inability of that component to demix from the other across a certain length scale. Okay, I think that's it for the questions. So let me just finish with one slide. And it's, again, a pitch why I think peptide condensates are really useful for the synthetic cell community. So the first point, I hope, is really obvious. It's a great way to create reactive separation media. If you want to spatial or temporally regulate some components, I've talked about partitioning of proteins. There's other groups that are interested in partitioning of small molecules. I think it's a really facile system for engineering. So we figured out rules so we can change individual amino acids on the sequence. That changes the saturation concentration, but can also change its viscosity. And then I showed you we can take two separate peptides that form discrete condensed phases, and then by changing a few of those residues, we can get them to co-assemble. So there's just a huge space here for controlling or engineering these to create synthetic organelles. And then I think I've hopefully convinced you this is a great way to prototype things. So you can certainly just take some of our existing modules or that other people have developed and start to think about linking them together. But again, I will just say one of the challenges for working with like TXTL and vesicles is that if you want to interface with something else, like another signaling system, well, that signaling system has to be there. And that's partly why we've been really trying to do this all top down using extant cells, right? That already have all these interesting capabilities and functionalities. And then we want to see if we make a synthetic organelle, can we now, push and pull the system in a way that's really predictable. And then finally, I think this is a really great paradigm for self-assembly. You can certainly create other types of rigid compartments from like nanocages. Protein design is so advanced now that you can create, things that are 100 nanometer in size, predictively from peptide self-assembly. But we've really found value in using these condensed phases themselves. Okay, so I will stop there and acknowledge the people who did this work. I want to really highlight Wental, Bobby, and Rachel, whose papers I've talked about today, and former members of the lab, a close collaborator, Dan Hammer, close collaborator, Jitain Mittal, as well as our funding. And I'd be happy to answer more questions.
Thank you. Thank you so much. I really liked it. Let's see, is there any more questions in chat? I don't see any, but there's something that it was sort of addressed by one of the questions, but I'm really interested in, we work with vesicles like a lot of people in our field is, do you think when you have those especially those really hydrophobic compartments, is there any chance to engineer some sort of an intrinsic interaction with vesicles? Something like we can use a vesicle surface to organize the compartments, but not messed up one or the other.
Yep. Yeah, and I didn't talk about this at all, but yeah, there's 5 or 6 great examples where people have basically targeted the co-assembly or the peptide assembly to occur on the membrane. And then you can get wetted patches, so more like 2-dimensional condensates. So these don't have to be, you don't have to make a micron size assembly. You can get that to be more localized to where you want. Gene Stakoviak's group has done a bunch of, nucleating assembly on membranes, seeing partitioning of individual lipids. And then I guess what I should point out is Ali's group has shown that you can actually get these HP pep particles to cross PC cholesterol vesicle membranes without macropinocytosis. And there's actually seemingly an explosion now in people doing this with other polymers. So just with like polyvinyl coacervates. And so I think given the generality of it, if you wanted to make a compartment outside a protocell and then have it cross that membrane, that's doable probably by a variety of strategies. And then if you want to use the membrane itself as a way to, create some type of a coacervate assembly so that you have a membrane and then a coacervate membrane. I think this is something your lab's particularly interested in. I think those things are all doable.
Thank you. Yeah, we had some experience with some of the IDPs, like the RGG domains, working with Chris Keating, and we had bad luck with liposome interactions. Like it seems like some of them were I don't really exactly know what they were doing. They were messing up liposomes in ways that made them look like they didn't want to form nice round vesicles, but instead they were assembling into weird bubble-like things. So I'm really curious to see how more of that work, because we need stuff that will be compatible. And that's why I'm excited about some of the stuff that you showed.
Yeah. Yeah, it might be worth thinking about that a little bit more. I mean, the first things that come to mind are that, the charge on the amphiphile or on the lipid is going to potentially recruit some of the stuff to the membrane. We've built these like RGG-based coacervates. They have a high PI in vesicles, and they're relatively stable, but certainly the type of amphiphile matters. So we mostly avoid phospholipids, honestly, right? We're use other types of amphiphiles. And so commonly we'll build things from dendrimers as ways to create a relatively durable membrane and to have kind of limited interactions with that interface. So yes, certainly there's a bunch of parameters that matter, but I think it's definitely doable to create a compartment that would not interfere with the membrane.
Yeah. All right, any other questions? Last call for questions. If there are none, I want to say thank you so much, Matt, again. And thanks for a great talk. And thanks everyone for a great discussion.
Really happy to be here. I love this community, and always fun to interact with everyone. All
right, have a great rest of your Monday. Bye.
Bye.